Good evening, children. How are you all keeping? I hope you all are keeping fine. All right. Today we'll start with our new cross. Last time I had introduced in genetics with the laws in genetic crosses. I had made you aware. We had learned the steps of making a Punnett Square that was introduced by the scientist Punnett, who had given certain layouts for making the square. Now, in the same context, I had told you, children, there were two tables, the summary of which I told you, please learn it by heart. The tables were dominant and recessive traits of some hereditary traits in humans, and the other there were 7 pairs of contrasting features of garden pea, those that were studied by Mendel. Now, without these two tables, children, by heart, nobody will be able to do these crosses that we are practicing, right? So please, this should be all in your mind, and nothing, no book and no other help should be provided. Okay, so last time we had started with the example 1 and 2 we did on monohybrid cross. Now, what was monohybrid cross? A cross in which the parents were with one contrasting character, right? There was just one contrasting character, that is, if one was tall, the other was dwarf, and the F1 generation was shown, the phenotype and genotype. The phenotype means the physical appearance of the progeny or the children born, and the genotype means the possible genetic combination. Then moving further, we introduced the Punnett Square, where we calculated the phenotype and genotype in F2 generation or the second filial generation. And did you notice, children, whether we do any feature or in a monohybrid cross of contrasting character, but the phenotype is always fixed in the ratio, that is 3:1 in F2, and genotype in F2 is 1:2:1. Then we did another cross and we justified what I am saying. Look at this answer once again. This was example 2 we did last time. I hope you've understood. Again, 3:1 with phenotype, and genotype came 1:2:1. Then a third question I gave you as an assignment. I hope you have done it honestly yourself, but the answer I had uploaded on the group, but I'm sure you have tallied. So here it was again the same answer. So with these 3 examples, at least this is clear that the method is a standard method and we will stick to this method. They are very simple. P1 or the first parental generation, gametes, and then F1. Then P2, again gamete by Punnett Square, and then F2 generation, that is the second filial generation. Now, today we are going to start one step ahead of our learning. Last time we did monohybrid cross. Today, let us start another very interesting part, and this is a dihybrid cross. Rules are the same, children, remember, but the things will be doubled. So what is a dihybrid cross, children? Dihybrid cross, I had introduced you. It is a cross in our previous classes. I hope you remember. Look here. It is here. The dihybrid cross, it is a cross in which we carry out by taking 2 pairs of contrasting characters at a time. These 2 pairs could be in any combination that we'll see in our cross today. So what is the difference between mono and dihybrid? If mono is one contrasting character at a time, here they will be 2. So let us see our question. The question says, a cross was made between 2 parents, 2 pea plants. One plant had round and yellow seeds, and the other variety had wrinkled and green seeds. Draw a Punnett square and show phenotype and genotype ratios of F1 and F2 generation. I already told you, children, rules are the same. Alright, so let us begin. The first step, as usual, P1 generation. We have to first choose our alphabets from the parents. So as I told you, children, without the tables we'll be helpless. So because today you have still not learned, so we'll use the tables. See, one parent has round and yellow seeds, whereas the other parent, where the cross is being done, has wrinkled and green seeds. So you're talking about 2 characters. One is the shape, that is round in one, the other is wrinkled. That is just the opposite, contrasting, correct? The other is with yellow and with green. So let us see the shape and the color into the tables. In the previous table, back to our table, let us see this. Seed, color and seed shape. So which color is dominant? Yellow over green, which is recessive, and round is dominant, whereas wrinkled is recessive. And the choice of our alphabets, children, is according to this table only. So what will be the dominant alphabet here? What will be the chosen alphabet? It will be R or W? Yes, you're right. We'll choose R. R for the shape, and yellow because it is dominant. Always remember, children, dominant or the super ruling gene has always a power to show or subdue the other, that is recessive. So R is one alphabet over W that we are preferring, and Y is the other over green. Why? Because obviously both are the dominant traits. So our alphabet selected: round, double R, round seeds, and yellow. I told you double Y. So let us write below. One of the parent is round and yellow seeds. Alright, and the other is just the contrast. What should it be? Small alphabets, because the other is wrinkled and green. We will not choose the alphabet W or G, but it will be the same alphabet RR, because the dominant will be given priority, and small y, small y. So this alphabet capital itself tells you that this is dominant, and small itself hints that yes, it is recessive. So let's write it below. This becomes wrinkled and green seeds. Same exactly, children. So coming to second step was the formation of gametes. And as I told you, the gamete formation is bringing it to half. Alright. So what will be the half of these doubles? Yes, it will be capital R and small capital Y. Right? Here they were double, you just bring it to half, or singles. Here also small r and small y. Second step over. Now coming to the third step, that is F1 generation. Now here you have to learn. In this, children, last time it was easy. We had just one alphabet, T and T, we brought together, giving priority to the capital. But this time we have two alphabets. Now what we are going to do is, we will keep same alphabets together. That is capital R, again a priority to be given, followed by small r, capital Y and small y. If you write it like this, children, there might be many chances of errors, like both the capitals together and both the small together. In this case, children, same alphabet is being split up. This will go wrong. So what we have to tell you, keep the similar alphabets together, no matter they are capital and small, but yes, priority always to the capital. So R, R, Y, Y. Now what will be the phenotype of this progeny? Phenotype and genotype. Phenotype, as I told you, the physical appearance. What will be a seed with this type of combination? Capital R means yes, it will be round, even if though it has one of the recessive parts here as the recessive gene, and this Y will be yellow. So all seeds are heterozygous. Remember heterozygous when capital and small alphabets are together, right? Unlike homozygous, where both of them they are same. Like this is homozygous, round and yellow, and this is also homozygous. You may write down homozygous condition. This was also homozygous, but this is yes heterozygous. So all seeds are heterozygous, round and yellow. Alright, now what will be the genotype? 100% capital R, small r, capital Y, small y. So one part of our question is over. Did you find any new thing? Not much, except that the things were doubled. I already told you rules would be the same and they will be just on one part, that yes, you are doubling them. So this is like the parents P1, P2, gametes, and then F1 generation, that is capital R, capital Y, small y. Now, coming to the second part of our work, we are going to choose now P2 parents. P2, that is the parents expecting that these children, when they become mature tomorrow, will be self-crossed. So who are our next parents, P2 generation? They will be the same. Capital R, capital Y and small y into same. Right? So like previous, this is the same thing like the previous one, only the children that were born in F1 were being self-crossed. Right? Here also the children are being born in F1 are being self-crossed. And now the next step, that is formation of gametes. Now this is what you have to learn, children. This will be a little elaborate method. Gamete formation in a dihybrid cross for F2 generation is a little elaborate work. See, look here. You need a bigger square. Because that time it was a monohybrid. All we needed were 4 boxes. But this time we have 16 boxes to create. So we need a big lot of work to do. Like see, this one was a very small square in P2 generation. Right? F2 generation, sorry. But here we have a big square. So let's see how we're going to do it. You'll do it by a scale and a pencil. 2 axes we'll choose. One is the horizontal, the other is vertical. Now you have to think of any possible combinations of splitting of these genes or segregation that can be possible. Alphabets would be only R and Y. Let us see. Right? So what we're going to do, you have 2 choices, R, Y, Y into R, Y, Y. Now how you're going to split up, it's very interesting, children. See, first let us make the boxes. 16 as I told you. Keep this for your heading and the first column for your heading, and then 16 boxes here. Alright. Now keeping the alphabets as both capital, like one of these you are bringing it on one axis. Right? One of these you will bring on this axis and split them. What all combinations can you make from this? Let's begin. First will be both capitals. See, R capital, small r, capital Y, small y. So how can you bring it into maximum combinations? Both capitals together, first one, and go to the last, extreme last, and make both small together, just the opposite. Capital first and the small characters or alphabets at the last. Right? Now what all possibilities are there? One capital, the other is small, capital R, small y, and small r and capital Y. This is the only combination. Think, children, any other possibility? No. So both the capital, you have done it. Both the small, done it. Capital and small, done it. And small and capital, again done it. Your work is being done. Now here you will choose another combination, but because it is in the same order and every time it will be, so choose the same order in the same priority, that is both the capitals, again same. Right? Here in the first order, see, both are now capitals, and I told you small at the last, so keep it small y, then capital R and small y, right, and small r and capital Y. Now before you have to proceed, please check that the order will be the same. Is it same? See, both capitals, yes, capital and small, yes, small and capital, yes, and both smalls. Now your work becomes like a game. What we are going to do, you have to just fill in the boxes, keeping 2 things in your mind, that filling should be in the order, give priority to the dominant gene, and yes, keep the alphabets together. So let us begin. Try it yourself with me. Both R, both Y. Both R, capital Y and small y. This R and this R and both capital. Right? Then capital R, small r, capital Y, small y. So what you're doing, children, exactly bringing one gene from here, the other from there, and mixing. Like see, one R and one R, Y and Y. So you're just bringing one pair here down and this pair, and whoever is capital, but keeping the same alphabet together. So capital being written first. Let's do for the second line. This R and this R, this Y and this Y, then both capitals and both smalls. Now here this is coming but will be written later because this capital will come first. So R, R, capital Y and small y. Then R, R, Y and Y comes like this. Coming to the third line, again capital first, small, both capitals. Capital R, capital Y, small y. Then here it will be both small, both capital. Then both small, capital and small. Finally the last filling up. Here you will be showing capital, small, capital and small. Alright. Then capital, small and both small. Then it is both the smalls, capital Y and small y. Then all four which are small. So this is the entire filling up for the dihybrid cross. Don't think your work is up. You still have to go with the phenotype and genotype, children. So phenotype ratio and genotype ratio. Right? Phenotype ratio, children, you have to do it in such a way that you're going to get 4 categories. Let us first see what will be capital R and capital Y. They will be round and yellow, because R, remember, we had done for round color and yellow was for dominant yellow color. So round and yellow, what is this going to be? Again round and yellow. What is this? Round and yellow, and again round and yellow. What will be this? Once again round yellow. This will not be, so this will be again round yellow. Even this won't be, so leave it. This is also round yellow, and this also is round yellow. See any other? Yes, this one. So round and yellow. See how many we have done. 1, 2, 3, 4, 5, 6, 7, 8, 9. So the first ratio is 9. What is 9? Round and yellow seeds. Alright. Is 2. Coming to the next category. What is this? R will be, yes, round, and double Y, which are small, will be green. So let's write it. Round and green. What will be this? Again round and green. Any other? This will be not round but wrinkled. This is wrinkled. Yes, this one. So this is round and green. So how many? Any other round and green? No. So the other combination of our choice is 3. How many are 3? What are they going to be? These are round and green seeds. Alright. Now coming to our last of these. Let us see what are these going to be. So this is round green and round green. If we number 1, 2 and 3. Alright. The rest have already been given for phenotype. Now what will be this? This will be, let us show by a diagonal work. This will be R small, means this will be wrinkled, and capital Y means yellow. So this will be wrinkled yellow. Again wrinkled yellow, and this again wrinkled yellow. So how many? 1, 2 and 3. So once again wrinkled and yellow seeds. Finally the one and only remains is this. What will be this, children? This will be the only single one, and what it is? Double R small means wrinkled and double small means green. So this will be wrinkled and green seeds. So what ratio did you get over here? Did you see you are getting the ratio as 9:3:3:1? And to know whether you are right or not, children, please add up. This should come to 16. Is it coming? 9 plus 3, 12, right? Plus 3, 15, plus 1, 16. So that means out of these 16 boxes that you have made, nothing has been missed out. And all you have done is that you've given the phenotype of all. That is, what are the expected plants going to look like? And what will be the genotype? You don't have to sit and do that. You just simply write the genotype would be very complex and leave your work like this. This is the complete answer to the first monohybrid cross. Once again, children, I hope you understood. This was very interesting. We had started with the cross called as dihybrid cross, where again we had two parts to calculate the F1 and F2 generations. And what was the dihybrid cross? The contrasting characters in two pairs, round and yellow. Because they were dominant, we chose the capital alphabets, being crossed with wrinkled and green, and they were recessive, so small alphabets. Same rules, we brought it to half, then made the F1 generation, priority giving to capital letter and keeping the alphabets together, ignoring this method where the alphabets would be split up, and then giving phenotype and genotype. And P2 generation, I haven't done it here because the space was less. So here P2 generation means again you have to form the gametes. Now you need the Punnett Square, which would be very big one with 16 boxes, how I told you, keeping one parent on one axis, both capitals and both last, capitals first, last at the end, and randomly write but in the same order, and fill up the boxes, categorize. Again your ratio, every time any number of crosses we do in dihybrid, children, your ratio will be fixed. Try doing yourself. 9:3:3:1. And genotype, don't worry, you have to just write very complex. So try this out with another cross today and write this as a homework with the same, but do it honestly, children. All you have to do is choose the alphabet wisely. In that case, if you miss, means you'd be wrong. Example 2. A brown-eyed man with normal vision marries a blue-eyed lady who is colorblind. Draw a Punnett square and show phenotype and genotype ratios of F1 and F2 generation. So once again, children, I'll just introduce the alphabets to you. P1 generation. Two things, once again, that means two contrasting characters. Let us see the table. Every time without this table you are not going to make your start with the work. Let us see. Eye color, brown is dominant over blue because both the alphabets are same, but you will choose B, that will be meaning brown for capital, right? And normal vision is capital over or dominant over colorblind. So what will be your alphabet? One alphabet will be capital B, and here it will be capital N. So just helping you with this, but the working children, try doing yourself. Capital B is brown-eyed into small b means blue-eyed, right? And normal vision, I told you, N with capital, so normal vision, and here again small n with color blindness. Once again, check it, children. Brown capital over blue, which is small b because this is recessive, and normal vision N will be dominant over color blindness. So this is your combination, children. Try yourself, same rules, bring it to half, form F1, then write the ratios, phenotype and genotype, and proceed with the Punnett square, how we've done it, and start with F2 generation, phenotype and genotype. But one hint I'll give you, yes, is that the answer rightly should come in the same ratio. Though I can give you just now, but I want you to try, and this answer we'll discuss next time so that you can try yourself. I hope you've understood, children. So today's part is still here. Tomorrow I'll be doing the other example and proceed further. Okay, thank you, children.